Welcome back to Hayden's Physiology channel. Today we're going to be talking about how smooth muscles, not the hard ones, but the smooth muscles work. So first and foremost, on our left here, we have this worm-looking thing. This worm-looking thing is actually an enzyme. It lives inside your body. Not a real worm, just an enzyme. This one is called MLCK. So this is MLCK in its inactive form. And then this over here is a different enzyme called calmodulin. So in order to activate MLCK, which we'll see later, contributes to the overall cycle of smooth muscle contraction, calmodulin first must receive calcium, 4 calcium to be exact, and then once it has all 4 calciums bound, our calmodulin is going to go and it's going to go and attack our snake-looking thing, and it's going to go from the inactive form to the active form. Now the primary difference between the two is this one has its tail in its mouth, this one does not. So this snake is ready to bite and do stuff inside the body. Okay, that's MLCK, not to be confused with its closely associated brother, who looks like Pac-Man, MLCP. So MLCP, we see it in 2 different forms. We see it in the inactive form and the active form. So the inactive form occurs due to another enzyme similar to calmodulin but has the opposite effect called, we're just going to draw this one like a square, called Rho kinase. So Rho kinase's role is, its whole existence is to inactivate MLCP. The way it does that is it attaches a phosphorus group. Essentially, MLCP is hungry for phosphorus, and when it doesn't have the phosphorus, it's going to go looking for it. When it does have the phosphorus, it's completely content. Hence, that's what Rho kinase does. Rho kinase, however, is instead of being activated by calcium, like we saw with calmodulin, it is actually deactivated with calcium. So as we increase levels of calcium inside the cell, we're going to see a more prevalent active form of it. Alright, so here we have our 2 different contraction cycles. This one we call phasic. So phasic contractions, we call them phasic because they exist in two different phases frequently, contracted and uncontracted. So starting over here, we have this is our actin Z-line filament, and then this is what we're going to call our myosin filament. So the main difference between skeletal muscles and smooth muscles is, we're actually going to call this is our myosin head, and we're going to have ADP and our traditional phosphorus as well, but we are going to have an additional phosphorus from MLCK, and we'll show that coming in later, MLCK phosphorylating the light chain. So when it gets phosphorylated, or we go through this process, and the myosin head is gonna form the cross bridge with our actin, and at that point in time, we still have our ADP, and we still have our phosphorus, and we still have the other phosphorus, the brand new phosphorus. Then we're gonna undergo the traditional power stroke, where the myosin head goes from its cocked position to its uncocked position down our myosin filament. But in that process, just like we do with skeletal muscles, we are going to lose ADP and phosphorus because we need the energy from their bonds in order to get this going. However, this is where it is different in the fact that we still have that additional phosphorus coming from MLCK. The point of this phosphorus is because we don't actually have troponin like we do in skeletal muscles. Actin is still bound to tropomyosin, so it needs some help in order to the myosin heads need some help in order to get to that myosin active binding site on the actin. That's what this phosphorus does, is it just gives the chain more energy, it makes it a more favorable reaction. So we go on to here, and after it's gone from the cocked position, 2 things could happen. Let's draw this deal here. So if the phosphorus falls off, let me draw the same step actually. Awesome. So it could happen in a way that the phosphorus falls off due to MLCP. If MLCP comes in. Let me draw that actually off. And, oh, you know what? That's actually a little backwards. Let me write that differently. Something wasn't quite making sense. Okay, trying that again. So then we're gonna have ATP coming in, and that is going to bind to our myosin head, causing our myosin to fall off of our actin. And that's gonna be ATP. Now here's where two different steps could happen. And we also still have our phosphorus still attached to our light chain. So it could happen that the ATP is just gonna hydrolyze and it's gonna go on, or the ATP is gonna hydrolyze and it's gonna attach. And this is called hydrolyze reattachment. Or the phosphorus could actually fall off due to MLCP in order to get that phosphorus off, and then MLCK will have to come in once again to add the phosphorus back on. Just two different steps. This step will happen if we have high enough amounts of MLCK and low amounts of MLCP, and we'll just keep going through our cycle. But if there's enough MLCP, we'll actually see that dephosphorylation and then the phosphorylation again in order for it to bind. Talking about what the concentrations of calcium, MLCK, and MLCP look like in terms of what's going on. So let's say blue is our calcium, so it'll go up and go down. And then let's say red is our MLCK, it'll also go up and sort of follow the trend of calcium. However, MLCP in phasic cycles looks a little bit different. It doesn't really go up that much, then it goes down. So that would mean we mostly see this, but occasionally we do get some MLCP that comes and kicks off our phosphorus, and we have to reset and add the phosphorus back on. Coming over here to the tonic sustained cycle, or the tonic cycle. Tonic, essentially it's like a one type of contraction, versus this one we can see either it's off or on. Tonic, we see a lot more singular contractions, meaning it'll contract and it just kind of stays on there. Because of this, we don't poop our pants, a wise friend once said. So, coming over to here, same deal. It's very similar, where we have our myosin head, we have our ADP, we have our phosphorus, and we also at this point in time, it's also been phosphorylated by our MLCK. And once again, our myosin head's going to come attach. And our phosphorus is there, and our other phosphorus is there. The phosphorus from the MLCK. And then we're going to do the power stroke again. Then we're going from cocked to uncocked. And again, ADP and phosphorus fall off, except for the phosphorus from MLCK. However, this is where it gets a bit different. So in tonic sustained contractions, we actually have a little bit more MLCP and higher concentration. So what might happen, because there's enough MLCP, and remember it's hungry for phosphorus, what might happen is our phosphorus might be taken off by our MLCP before we get to before the myosin head can fall off the actin filament. That causes not really an issue, but an issue for the myosin head, where now it doesn't have enough energy to actually fall off, and it's going to stay in that contracted state. This is just a really efficient way for muscles to stay contracted without actually consuming more and more energy. This phase we call the latch stage, because it stays latched. We're not going through a phasic cycle like we saw with the phasic muscles. For concentration levels of our calcium and phosphor, or calcium, MLCK, and MLCP, sticking with our color scheme, just kidding, we messed up. By we, I mean I. So what actually happens here is so we do go through the cycle, but when we go from our cocked to our uncocked, MLCP comes in at this point, and it is actually going to take off our phosphorus. So we're gonna have a bare myosin head. Now what is true is that it is stuck in that position and so once the ADP, phosphorus, and the phosphorus from the MLCK have fallen off, again our myosin head is kind of stuck in place. That's gonna make it really difficult for ATP to come in and attach to our head here. It's going to try, but it might not succeed. What it does, it finally will hydrolyze, but this process of it trying to attach, in the meantime, we're stuck in this latch phase, that phase where it's attached to our muscle in the contracted form, which means our muscle can't uncontract and we get free contraction, meaning we're not constantly unlatching and latching. We're not seeing that phasic cycle, we're seeing a tonic cycle. Going to our concentration levels, so we have our calcium. Our calcium is going to rise and stay more or less constant. Our MLCK levels are going to also rise, make sure it's right, and they're going to stay increased. And then same thing's actually going to happen with our MLCP. And we see a similar trend throughout them all because when MLCK and MLCP values are equal to each other, that's when we see this, oh crap, there's not enough, we're not seeing enough phosphorus from MLCK on our light chain, and that's why it gets stuck in the first place. Remember, this is actually a good thing. Last thing to talk about is this deal. So where we get our calcium from. So on this side we have the first way we can get calcium from our intracellular fluid is through phospholipase C. So phospholipase C is just actually a surface receptor, and we'll have some sort of ligand, something, come and bind to this, which is going to cause an IP3 cascade. I said I, it's supposed to be P. IP3 cascade to come and attach and allow calcium to flow out of the cell, hence we have intracellular calcium. Ligand gated and voltage gated, those are going to allow calcium to come from the extracellular fluid and come into our cell. And stress activated receptors, that's a response where we can activate our phasic cycle and just further activate our ligand and voltage gated receptors depending on like how our muscles are stretched or how they're not stretched.